I'm Kevin Elmy. In this presentation, we are going to talk about changes in soil biology. Our soils are a very dynamic ecological system. They are so very fragile, yet they are so adaptive. They are so complex, but yet so simple. Our soils have been developed over centuries. They've been put through drought, flood, fire, locust, erosion, glaciation, everything that the climate can throw at it, our soils have seen it. And they've adapted and to be able to grow plants under all of those conditions. Our actual soil has a very intricate relationship between plants, animals that have been grazing on them, the soil biology within it, and the parent material or the rock material that has used as the base of our soils. All of those really influence what kind of soils we do produce. And when you look at the history of mankind, the most damaging invention, as Dr. Montgomery has said, is the plow. That has made the most damage. It's changed the soil biology dramatically. It's gone from a natural system to something less than natural. And farmers have been waging war against nature and plant ecology right from that day that the first plow hit the ground. And when we start this, stop the natural soil evolution and the plant ecology, that's one thing. We need to keep it in there to keep growing annual crops. But the problem with our ag system right now is we tend to hit that reset button too hard, and we hit it too often. What I mean by that is, in this war of mankind versus nature and having plant succession, soil ecology build, is nature wants to progress toward tree landscape. When you look at the Canadian prairies, prior to the European settlers coming here, there was basically wide open prairies, very little trees, and herds of grazing animals going through and grazing and migrating and doing their thing. And that's happened for centuries. When European settlers came and we started putting in rail lines, we put in roads, those are natural fire breaks. And what would happen is if the grasslands, if it progressed too far to the right, as in that image that's up, we get a fire. Well, that moves that plant ecology or plant succession to the left. So then it moves it back from shrubs and trees back to the grasses again. And so that's been happening, quite regularly through history. When the plow came in, now we keep moving it to the left. We don't allow it to keep moving to the right somewhat to build up some fungal component in our soils, and it keeps moving it back. So my quick rule of thumb is most agriculture, we would be best off if we were in that zero point 0.5 fungal to bacteria ratio, and that ratio is based on pounds per pound. Modern conventional agriculture right now, we are well under 3. We're being that 0.15 to zero point 0.2 fungal to bacteria ratio. So that tells us that our soils are dominated by bacteria, and bacterial dominated soils are addicted to inputs. So if we want to get away from these inputs, we have to build fungi in our soils. So what changes these ratios? So when we start looking at tillage, the use of synthetics, using of residues with a tight carbon to nitrogen ratio, all of those promote bacterial growth. When we're dealing with manure, it's going to be one of the variables in there is, are we talking about straight out the animal, which has a very tight carbon to nitrogen ratio that will promote a lot of bacteria. When we're including the bedding and the straw, depending on how much straw is in there, will influence how much bacterial growth versus fungal growth. So the more straw, the harder the residue is to break down, the more fungal dominated will occur in that residue. So when we start looking at what we feeding the soil and in the what we're feeding the microbes, anything that is easy to digest and break down, that causes quick growth in the bacteria population. The fungi are able to use those nutrients, but they're really slow moving and getting to them, so most of the time they miss out on it. When we look at more complex food sources, so your lignins, uh, some of those longer carbon chains that the bacteria can't break down, the fungi are able to break down. Takes a longer process. That's part of the reason why mob grazing in a holistic system works. So in this system, we're going to produce, if we're going to allow those plants to fully recover, they are going to produce a lot of above ground biomass, which then drives a good root biomass also. That above ground biomass, it's going to produce something with a relatively wide carbon nitrogen ratio. So that's going to feed our fungi. There's no tillage, which then once again supports a higher fungal to bacteria ratio, because with tillage breaks up your fungi and decreases your populations because they have to regrow. So no tillage is a positive. When we look at a diverse plant population, those diverse plant populations are going to have diverse root exudates, which is going to give a diverse soil microbiology in that soil. Then we're allowing that plant to fully recover, have that root system fully recharge before that next grazing occurrence is going to happen. So that is giving you your continuous living root in this system. Jay Fuhr at the Mannequin Farms, he'd pulled up this chart, this research chart that he did off of the farm. And his quick rule of thumb is when you're grazing, ideally you graze the top half and you leave the bottom half. And this, when you look at some of these numbers, it's fairly obvious of why we want to do this. Anytime when we start grazing animals and we're consuming that bottom half of the plant, yeah, we're using up all the feed out there. But when you look at the feed values of that bottom half of the plant, your relative feed values are lower, your protein is lower, your total digestible nutrients are lower, and usually that's where you're going to have any of your issues of feed tests. So your nitrates, higher sulfates, higher lignin, all of those things. So the way that you can use that as a gauge is if you take a plant and you cut it off right at the soil surface, and then you balance it on your finger, and that is your 50%. So it's 50% in weight. So the bottom half of your plant is going to weigh more than your top half because of lignin and a thicker stem, all this other good stuff. And so when you balance it, it's not going to be in the middle of the plant per se. It's going to be, the bottom third of the plant, but that is half of the weight. And that's the quick, easy, dirty way of going in there and finding out where half is. When we have that armor on the soil surface, not only is it giving the soil protection, leaving the low relative feed value on the soil surface, but it gives that fungi something to eat. That's the stuff they're masters at breaking down. When we go through and we're doing this grazing, so we have this fungal growing fungal to bacteria ratio happening. When we go in, we start grazing it. Now that manure and urine that the animals are going to be dropping is going to promote more bacteria. So that's going to help cycle nutrients. So your bacteria cycles your nutrients very quick. The whole thing is if we go in and we do something like swath grazing where we don't have that living root, we have relatively high stocking rates and we have a lot of manure on the ground. Now we've produced a lot more bacteria in that soil system, and that bacteria will allow nitrates to accumulate. When we have excess nitrates in our system, in our soil system, this is where we have early successional plants starting to get their foothold and start growing. When we have fungal dominated soils, what happens is most of the nitrogen that is available to the plants is in the ammonium stage. Ideally, we'd like to have a little bit of balance of that, but in the big picture, that's kind of the quick nuts and bolts about that, the system. So by having that balance of fungal to bacteria ratio, the forage plants still need access to some nitrates. So we're not getting rid of all the bacteria because we need that for those nutrient cycles. But when the bacteria population gets too low, so if we are overgrazing, so if we're not allowing that plant to fully recover, if we don't have those stocking rates that we need, if under Alan Savory's holistic view, when he talks about partial rest, it took me a little time to get my head around that idea. But if we had a quarter section of land of pasture and we don't have that broken down into paddocks, that partial rest means that the animal can go in, graze anywhere they want, and they're going to graze the stuff they grazed. They're going to want to graze the fresh growth first. And by doing that, they're going to allow some plants to rest and some not. And that's part of what he means by this partial rest, which, like I said, it took my brain to get around that one a little bit. But so when we're dealing with this bacteria population, we have this pasture that's been overgrazed, undergrazed. What happens is we don't have that manure, we don't have that hoof action. Our bacteria population starts to get low. And if our fungal population is still healthy and still doing its job, then our fungal to bacteria ratio starts getting to be in close to that 1-to-1 or even higher. When that happens, our bacteria numbers are dropping, our production in that field starts dropping, and then we start seeing woody species starting to appear in that pasture. That ratio is just a ratio. To give a quick guidance, we need the absolute numbers to see when our productivity starts dropping, where's our actual numbers. And so that's where we're doing some testing is one of the rules of thumb. If you measure it, then you can manage it. So in all ecological systems with no system shocks to it, that system will revert to the lowest energy level. That lowest energy level means it's going to be low productivity. This is where that holistic grazing supplies that rest, allows that plant to fully recover, and then comes in with a shock with a high density grazing. You get the manure, you allow that full rest. That system then is sustainable. When and if you've heard me talk before, you've seen this graph, and this just goes through the different fungal to bacteria ratios. And so down in the bottom where we're dealing with purely bacterial soils, that's bare parent material, then we start getting more and more fungal into it. So that when we are looking at this, and we're in that 0.1 fungal to bacteria ratio, this is when we start dealing with a lot of weeds. They talk about Johnson grass, but high nitrates, lack of oxygen. And that lack of oxygen, we'll talk about it in a little bit. When we start developing good fungal communities in our soils, one of the things that the fungi is able to do is take soil bound calcium and change it into calcium oxalate. That calcium oxalate is highly plant available. When that calcium is in that calcium oxalate state, now we're going to get better soil structure. So is it because of the fungi? Is it because of the proper form or better form of calcium in the system? It doesn't really matter. But anyways, as we start increasing the fungi, we start getting more and more diverse plants starting to get established. So when we're in that under that 0.3, that's when we have the high nitrate. This is when we have the weeds. This is when we have the lack of oxygen. We have poor soil structure. We get over that 0.3, now we start seeing granulation. When we get into that 0.75, into that 1-to-1 ratio, that is where we want to be with our grasses. When we look at agriculture, we want to be under that 0.75. So, in that 0.5 to 0.75 is kind of where I would say the sweet spot would be. When we get over that 1 to 1, and you'll see the shrubs, vines, bushes, things like that, once again, we need to find out where we are with our numbers. So if we know the ratio is high, and if it's that 2 to 1, for instance, so we're getting a little on the high side for having too much fungi. Where are we? Are we at a 10 to 5, or are we at 100 to 50? Because when we have more biology active in our soils are going to be more productive. So it's not going to be as much of an issue. Whereas if we're at a 10 to 5, well, we have to drive up both those numbers. And so when we change and we start managing to get more bacteria in our soils, that's going to change that ratio dramatically very quickly. So that's one of the things that we really have to be cognizant of. So from that slide, the fungal to bacteria ratios will influence what plants will grow. Basic plant succession. When we have a low fungal to bacteria ratio, that's early successional plants. We regard those as weeds. If we go in there, and in this case, this is one of my fields this year that my neighbor called the weed inspector on me because he saw all those weeds out there. If I would have gone in there, if I would have sprayed it, if I would have worked it, if I would have done anything with the traditional management, instead of allowing my fungal component to be increasing, to allow the species that I seeded to get established, I would have nuked it. Once again, press that reset button. Instead of going over 0.35 and going to 0.4, I would have nuked it, and I would have been back to 0.15, 0.2, somewhere in there. Guess what? Same weeds would be coming up again. I keep asking whether it's conventional or organic producers, that if your management of the weeds that you have, and these weeds just keep getting worse, you just can't get rid of them, maybe the system isn't working that you're trying to manage them. Maybe you have to look at a different way. And this is where just increasing that, having that living root in the soil, in the vegetative stage, increasing amount of fungi that is being built in your soil, those get rid of all of our early successional plants. We'll talk about some of the other perennials in a little bit, but that is basically what we want to be looking at, is increasing this fungal to bacteria ratio, getting away from that 0.2, 0.25, get into that 0.35, get into the 0.5. Then farming becomes fun. Now we get away from all these weed issues that we see. So as our fungal to bacteria ratio increases, ranked successional plants are now signaled to grow. So if anybody has tried to grow Sicer milk vetch, you'll seed Sicer milk vetch into cash crop ground, and normally you do not see that plant for 3 years. That first year, it's kind of there, it kind of germinates, maybe it kind of rosettes up, but the reason why it doesn't take off is it wants to have a fungal to bacteria ratio about a 0.5. So if we have it into a pasture situation where we have it seeded into a pasture blend, those first 3 years, you don't expect much. As that fungal increases, then we start seeing it. So if we had a field that we were able to take and develop a good fungal bacteria ratio, then add it, it would establish way quicker. This goes for things like any of your native species, so your green needlegrass, your western wheatgrass, your northern wheatgrass. All those species, if you put them in a blend, for the first few years you don't see them, because they need a wider fungal to bacteria ratio. They need to have something over 0.5. Once we get there, they take off, they do well. That's the other thing about when we start grazing native pastures, they need to have a high fungal bacteria ratio. So as soon as we start abusing them and increasing the bacteria content too high, they start disappearing. So these are the, once again, signals that we want to be looking at to be able to manage how, what plants do we want to see growing. When we look at the nutrient unavailability, that messes up succession. So when we have low calcium, and when we have low calcium, that means our fungal populations are dropping. When we have high nitrate, that means that we have our bacterial numbers starting to creep up. Or we have high sodium. Once again, that's not a soil thing per se, it's more of a water issue. So we're not managing that soil water efficiently. When we have those nutrient interference, that then moves us backwards. So how do we get around that? And that's going to vary from case to case. Succession then battles with ecology, because when we have these issues, what plants are best adapted to fix that soil? A lot of these weeds that show up have ecological advantage over the crops or the plants that we want to grow. Once we've identified what these weeds are, now the simple way to replace them, get rid of them, is pick species that will replace those weeds. They'll do the same job as those weeds. And one of the books I highly recommend for producers is a book by J.L. McCammon, When Weeds Talk. Great little book. Goes through, gives some really good information about some of the management practices you can be doing. And then in the middle part, close to the back, it has a nice chart where it lists a lot of the common weeds. And what it'll do is it'll tell you the common name, the scientific name, and then it has a bunch of cells where it shows different nutrients and what imbalance that weed requires to have its advantage. Great little book. I think it's about 20-25 bucks. Acres USA. I've seen it on there. A real good reference book to have. So some of the examples, we'll go through a couple of examples of this. Canadian thistle is one that usually pops up that people have issues with. And when you look at the book, it's actually page 95 is if you, if you have it, you can take a look. But basically, what we're looking at with Canadian thistle, it's an indication that you have anaerobic soils. So that tells us that we have low calcium, we'll have low P, low phosphate, low calcium, low humus, low porosity, so hard soils. Those soils would be highly bacterial. It has a deep root system. And what the thistle is trying to do is trying to open up that soil and cycle calcium for us. When we catch thistle early enough in its growing season, we can graze it, and the animals do extremely well on it before it gets its spines. One of the plant ecologists that I was talking to said that anytime a plant needs to have a physical defense to protect itself against something eating it, that means that that plant is full of nutrition, which kind of makes sense. So with Canadian thistle, okay, if those are the conditions that it likes, so what the way we get rid of Canadian thistle is do the opposite of what it likes. So it likes low calcium, so let's make more calcium and phosphate available. It likes anaerobic conditions, so let's aerate this soil. Let's increase the fungi. And the really neat thing with all of those four is they're all related. Once we start increasing the fungi then create soil structure, which then creates aeration. It makes that calcium more available. With especially the mycorrhizae has a bacteria that associates with it that's a phosphate solubilizer. So all of those four things all go back to trying to get that fungi happy. First of all, we need to be able to crack that soil and open it up. And that's where seeding an annual plant, or maybe a biennial or both, with a deep taproot. We need to grow. We need to drill down with its root and then die. When it dies, that root then dries up, shrinks down. Bingo bango, we have a macropore in our soil, which is allowing air to get in, creating aeration. Ideally, bringing up calcium in a tuber like the radish makes things that much better, and it's even better yet if it is mycorrhizal friendly. And that is the problem with using any of the brassicas. They're not mycorrhizal friendly. So using something like sweet clover, using something like chicory, throwing in a grass in there, these are the steps that we want to build fungi in those areas. A lot of people will go in and say that to get rid of thistles, you need to deep rip. When we start deep ripping, that goes actually the opposite direction, because deep ripping and doing lots of tillage will break up your fungi, so decrease your fungi numbers, and it'll stimulate bacteria. And if you've ever seen a slake test done, when we have bacterial based soils, those bacterial based soils, the structure collapses very quickly. So we can go in, do uh, roots, not iron. We'll be able to fix this problem significantly quicker and keep it away for a longer period of time. So the radishes, like I said, are a good candidate for a single shot. Uh, Basically, we want to seed them relatively early in the year, so that this way maybe we will go in and do some tillage and knock back the thistles. Seed these at a fairly high rate. What we want to do is watch that patch, so that anytime you see a thistle getting ahead of the radishes, we want to go in and we just want to clip the top of the radishes and make sure we clip those thistle tops off, because then they're going to have to regrow. When they start regrowing, that weakens that root of that thistle. Whereas as long as we leave enough of the radish leaf, it's not really going to get hurt. Once again, just like Jay's rule of clipping 50% or grazing 50%, this is going to be the same thing with these thistles and the radishes. If we can leave at least 50% of the radish leaf intact, they're going to regrow faster than what the thistle will. And that's where, the next step in this is adding in a little bit of chicory, or a little bit of oats or something like that, just to get more diversity. So that radish brings up an incredible amount of calcium. The calcium in plants, a large portion of it is located in the cell walls. So something that has more cell walls, so something that's bigger, is going to be bringing up more calcium. That calcium, when that root rots, is going to release, get into the soil, flocculate that soil, create macropores, feed that soil biology, and it takes away that Canadian thistle's ecological advantage. Example number 2, Foxtail, Barley, and Kosher. So that's, both of those, they grow in areas with high sodium, high potassium, bacterial dominated, high magnesium, high nitrates. So we're dealing with hard soils. Anytime you're dealing with low calcium, high magnesium, and bacterial, that's just a recipe for basically concrete. And in those areas, you'll have high nitrates. Once again, goes hand in hand with bacterial dominated. The calcium's going to be low, the phosphate low, and you're going to have low humus. The foxtail barley will grow in wetter areas. The kosher will grow in drier areas. And this was a picture, I think, from 2011. It rained a little bit that year, so our water table is a little high. And after the water started going down, this is what we started seeing on our farm. Just foxtail went crazy. So both of them, both the foxtail and the kochia, like those saline areas, hard soils. Both of those are trying to mellow up that soil. Once again, they're trying to help us out. We need to find ways of finding species that are going to replace them. In those areas, we want to increase the calcium, create macropores, internal drainage, increase our fungal component. We want to tie up nitrates, tie up that magnesium, and leach out that sodium. And that is, once again, we need those macropores. We need those pores going into the soil surface. And that's the reason why, if you have a penetrometer, we want to see where that hard pan is. where it is, and then manage it. Open it up, get those roots down, get our soils functioning again. Once again, tuber crops, really easy way to bring up calcium, ties up the nitrates because all of your brassicas are scavengers. In this case, this is a picture of a beet. It's not a brassica, but I still use them in the broadleaf group. Neat thing with the beets, this is the fodder beet. Neat thing with it is flea beetles do not like it. I haven't seen any numbers coming back with clubroot, so a real nice addition into a lot of these mixes. Negative is it doesn't have a lot of drought tolerance, but definitely is an option in mixes. They produce those macropores and get something in with that deep root, creates a macropore, leaches the salts away. Wild oats and cleavers. These are a little simpler to get rid of because basically that just shows that you have a low fungal to bacteria ratio. It shows you have free nitrates, low calcium. Tillage and fertilizer and spraying drives wild oat and cleaver growth and population. So the more tillage we do, the more fertilizer we use, the more herbicides we use. As crazy as this sounds, the more wild oats and cleavers are going to have. Because what we're doing is supporting that bacteria population. We're not supporting the fungal portion of it. Tie up the nitrates, wild oats disappear. Unwanted early successional plants, aka weeds. Once you know their ecological advantage, it's easy to deal with. And as a quick rule of thumb, keep your calcium active and cycling. Build the fungi in your soil. If you can keep that fungal to bacteria ratio in that 0.5 to 0.8, every action, every crop we grow, everything we do to manage is going to increase or decrease that. The whole idea of when we start talking about intercropping, when we start about cover cropping, when we start talking about relay, all of these different managements, that is going to counteract some of the stuff we did during the rest of the growing season. By doing that, we can now choose and influence what plants we want to be growing. Some of the tools we can get in there to get to that next level. If the cover crop, if things aren't fast enough, you want to get there faster, of soil health and balance in our soils. Now we can start talking about the use of compost. And this is once again more potential for directing some of the microbial balance in our soils. And so the three main types of compost that we can talk about is your dry compost, your compost teas, and your compost extracts. Your dry compost is going to be your bulkiest, so you're going to be taking a recipe and creating these piles, whether you're mixing them or your static piles, and then you're going to be putting them on your field. That's going to take a lot of biomass. When we start looking at the teas, now we're going to take some of that dry compost, put it into water, and brew it to create the biology we want, and then we're going to either put it in furrow or spray it on. When we start looking at extracts, this is going to be the most concentrated part. And once again, Jay, if you listen to some of his stuff, he has what he calls his worm juice. And so you'll have a 1,000-liter tote. He'll have his compost in these totes. He'll have earthworms in there actively doing their things, and you have to keep them fairly moist, so he keeps adding water. On the bottom of the tank, he has open, and out in the bottom comes this compost extract. So it's worm juice. It's some of the compost liquid that's coming out, and that is very concentrated. And what he found was if you tried putting it on full rate in furrow, you'll kill your plants because the microbes go crazy on it and they don't pay attention to the plants. So that's part of it. So it's very, very concentrated. So depending on what scale and how much work you want to do, those are your three options. Your fungal to bacteria ratio of each one of those products is going to be determined of what you used as your food source in your compost. So if we're using more wood chips, wider seed end ratio, we're going to get more fungal. If we have something that is greener, more lush, tighter C to N ratio, we're going to get more bacteria. So depending on what we want to be putting on, that's going to influence what our end product is going to be. It does take a lot of labor. It takes a lot of time. And it takes knowledge. You can be having your compost pile, and you could be doing everything right, and you miss one step and you can have basically garbage, worse than garbage, because you it can't get to the point where it wouldn't be safe to even put it onto your land. So that's one of the things. Make sure you do all your homework, start small, make sure you're doing everything right as you go along. The other thing is, when we're putting on these composts or extracts or the teas, we have to be putting them on basically living plants, because we're putting biology in the soil. We have to have some reason for that extra biology putting in the ground to survive and thrive. In John Kempf's book, Quality Agriculture, awesome read by the way, one of his interviews with Tom Dykstra. Tom is a consultant in the States, and one of the things that he talks about is when he has one of his clients with an area with poor plant production, he recommends that they go in and add 1 pound of sugar per acre per week and just keep going back. And when you have weeds starting to grow, mow them down, put on another pound, and doing that for a season. What that's going to do is it's going to feed your bacteria. And once again, is it sugar? Is it molasses? It doesn't matter. Just some simple food source to feed and build your bacteria numbers. When we start building our bacteria numbers, they're going to create those rasta roots. So those roots where they have all that soil stuck to them, they're going to be, have, they're going to live and they're going to die. They're going to produce glues in the soil. They're going to start that soil structure. And when they get the soil structure, you have the sugar, you get the nutrient cycling, then you're actually going to start getting plant production. When you get plant production, now we can start building fungi. So it's a real neat concept. Haven't heard of anybody doing it in Canada yet, but it definitely has a place where it definitely has a fit. The other ones you can take a look at are your humic acid, your fulvic acid, and biochar. It's another way of adding a carbon source into the system. And when you look at all carbon-based life, all nutrition that goes into us is always attached to carbon, minus water. So when we're adding fertilizer to our systems, we should always have a carbon attached to that fertilizer to help improve uptake and utilization. The other really neat information in there in some of the books. What book is this? I can't remember. But 1 ton of nitrogen fertilizer will burn off 30 metric tons of carbon out of the soil. I didn't realize it was that aggressive. So once again, when we're looking at rotations, when we're adding, all these different supplements, when we're grazing, keeping that nitrogen in check is really, really important. So with your humic acids, it's a long carbon chain. It's a chelator, and it's best that you put humic acid in the soil. Fulvic acid is a shorter carbon chain than the humic acid. It's a weaker chelator, and that's best to either put right on the seed or do foliar. I didn't have a picture for it, so threw the dog into it. Biochar, it's basically incomplete burned carbon. So it'll be, ash of some sort. And what'll happen is when you put it in the soil, it'll help create soil structure. So in those areas where we have thistles, where we have Foxtail barley or Kosher, you put biochar in. So this picture of this soil, you can see there's no structure to it. We throw some biochar in there, and that'll create instant structure. But I'm not sure on how it fits in broad span Canadian prairies. It takes a lot of energy to produce, and from the books that I've read, it works best in warm, humid climates, just because it's not going to get eaten up right away. So this way, it goes in the soil, creates that soil structure, and it's a very slow release. Soil sampling. I love this presentation because it's really feeding my ADD. I'm able to bounce all over the place. So the question I ask is, what are you testing for? And so that is going to then determine what depth you need to sample and how many samples you need to take off of a field. For nutrient testing, 2 to 3 average spots is all I use, because my rule of thumb, the way I've been trained, is I can pick 3 good spots off of a quarter section. I cannot pick 15 unless you're actually going to go GPS mark them and go around. But if you pick 3 truly average spots, you get a really good idea of that average on that field. And if you have a spot that is poor or troubles, grab a separate sample from that, get that sampled separately, because you're going to use those 2 or 3 average spots as your baseline. Then when you get back the soil test, you're going to be able to take a look at, okay, oh, this is the problem. This one's really different. So this is what the issue is. When we're looking, well, I guess going back to this, when we are looking at nutrient testing and we have our systems working properly, we'll get to a point where our soil test will show up extremely low everything. And that's because if we have a growing plant, a living plant, living root, cycling nutrients, it's going to be tying up these nutrients. And when they die, they're going to release it. And when they release it, that next plant's going to be growing and it's going to tie up those nutrients. So our soil test may show us really, really low levels, but we'll have really high productivity. And this is where testing for biology is going to be one of those things that will give you that idea. So if you have really good biology, you have you're using the 5 principles of soil health. Bang, our chemical soil test will show that very little is in it. The soil biology, each lab will have its own protocol. The Chinook Applied Research Association, uh, they have their protocol, and what they say is if you have a shallow compaction layer, so in that 3-inch range, then you want to take 2 samples, so 0 to 3, 3 to 6, and then you can just take a 0 to 6 if that compaction is deeper. If you just don't know where that compaction layer is, if it's deeper than that. I prefer to use a square shovel when I'm doing my soil sampling. So what I'll do is I will dig a hole in the soil. Then what I'll do is I will take basically a slice of bread along the face of that hole, and that's my sample. So then on that shovel, I pick it up, I take a look at it, I smell it, look for roots, look for bugs, look for earthworms. I examine it, because I want to see, okay, is this an average? I think this is an average spot. I look at it, and maybe there's some salt flecks down at 8 inches. Oh, okay, I got to remember that. So it just taking a look at it, that's going to be one of the biggest things to do. Once again, when we start looking at this complexity of the soil and what we're trying to do, there's a whole bunch of really neat diagrams trying to explain it. And this is the stuff we know. And if we're vague enough in each one of those arrows, we're going to be right. We don't know why we're right, but when you look at this, some of those boxes are really vague, and it doesn't matter. It just shows that there's a lot of things happening. And so to stay on top of it, you go crazy if you end up with a haircut like me if you want to try and get to the nuts and bolts of it. But you know, basically, just have to remember that the soil dynamic ecosystem, and we cannot and should not see, and we may not ever understand all of it, because there's viruses. Where do viruses fit in the soil ecology? We don't know. We're starting to get some of the research, but it's going to be a long time before we'll ever get that figured out. But the biggest thing with it, when we're looking at changes in the soil biology, is take a look at these simple observations to do. Number one, every once in a while, dig a soil pit, and that may be if you have a dead cow and you're going to go bury it. Get the backhoe out, dig before you throw the cow in. Get into the soil pit and take a look to see what you're where you're going to be burying the old girl. If you're going to be water lines, anytime you can, get into the soil, and how deep is that A horizon? Do we see any roots, where the roots are start growing sideways instead of going down? Soil testing, it is important, especially when you're starting out, to find out what are our issues, where's our balance, whether it's the nutrients or it's the biology part. Take a look at your plant growth. How are these plants growing? Are they stressed at a certain time of the year? And that may be because of the roots get down to that hard pan and they're trying to get through, but they just can't. They're fighting through it. And the other one is observe your weeds. Those weeds will tell you what is happening in the soil or what's not happening. Really good indicator of what's happening under the ground. Couple of nice little slides here. Everybody is the plants and the animals credit on building soil, and there's fungi doing everything. The other one, this must be my agent. The phone's ringing. I got a gig sequestering carbon. Without animals to sequester carbon, build soil, really really tough. So that's it. If you have the opportunity, if you're reading the book, I hope you get some benefit out of it. And if you have any questions, feel free to reach out to me, and we'll help out going through this as much as we can.